So let's talk about the model 3 of BCHEM 563 or BCHEM 565. Okay, and in this one I'm going to look at post-transcriptional regulation in eukaryotes. Okay, so regulation, or let's see, regulation is that okay after transcription. Okay, so this initial pages, we've already talked about them in the previous lecture about the differences between RNA and DNA. Okay, but what you have to know is RNA is actually it's usually single-stranded, unlike DNA, which is the double helix, right? But in its normal form, RNA can form it can be paired with each other. So probably if there are any complementary bases within the single strand of RNA, they can be paired. Okay, so it can form secondary structures, but it's in its primary form, it is actually a linear molecule. Okay, so unlike DNA that strictly follows the Watson and Crick base pairing rule, yes, RNA follows it as well, but in its free state, when it is forming the secondary structures, U can also be paired with gene. Okay, under normal circumstances, U be paired with A and gene with C. Okay, but when the single strand is just coiling up or bending to form be paired with other part of the same molecule, gene can be paired with U. Okay, this does not occur during transcription. It is only during folding. Okay, and this one too, I'm sure you know how the nucleotides are formed. It's the same as how the deoxynucleotides are formed. Okay, this is ribonucleotides. So in place of T, we will have U. Okay, so now let's look at the classes of RNA. Mainly we have two general classes: the coding and the non-coding, or you can call the coding and functional. Okay, for the coding RNA, it is just the mRNA, the messenger RNA, because it is the one that carries the genetic information from DNA to be translated. And the functional ones are tRNAs and rRNAs. Okay, so what they do is they play a role in the transfer of information from DNA to protein, and I'm sure we all know the role they play in translation. Okay, so tRNA, we know the role tRNA plays in translation, and we also know the role rRNA plays in translation. There are other functional RNAs as well, and those ones play a role in processing of other RNAs. Okay, the snRNA, snORNAs. Okay, and some also regulate RNA and protein levels in the cells, like microRNAs, pi and psi. We will look at them as well. Okay. So I'm sure you know about mRNA, tRNA, and RNA. So let's look at the other functional RNAs, those that participate in the processing of RNA and other RNA that are specific to eukaryotes. Okay, so I don't know if I've mentioned it, but small nuclear RNAs, snRNAs. Please forgive me for the background noise. So the snRNAs form a part of the spliceosome. Okay, spliceosome is the machinery that carries out splicing, cutting of introns from exons. Okay, so snRNA is part of the spliceosome. So we attribute the splicing of mRNAs to snRNAs. Okay, so it takes part in splicing mRNAs. snORNAs, that's same, but in this case not on mRNA molecules, but rather on tRNA and rRNAs. Okay, and they also guide chemical modifications of these same molecules. Okay, so they help splice rRNAs, tRNAs, and they also help in chemical modifications of these RNAs. And there are some that also help in regulation of genes. Okay, so I think the points are here. Some are the processing ones. The processing ones are what we just talked about, the SN and snORNAs, and those that regulate RNA and protein levels are the microRNAs and small interfering RNAs and the PIWI interacting RNAs. So let's look at the microRNAs or the miRNA. The miRNAs, they just play like they have a widespread role in regulating the amount of protein produced in many ways. So this is not a bit more direct. Their role is not really stated, like what exactly they do, but what you have to know is they play a role in regulating protein synthesis or protein expression. Okay, so they regulate the amount of protein produced, okay, in many eukaryotic genes, and they're actually tissue specific. Okay, so they do not function in all kind of genes. And looking at the psi and pi RNAs, that's the small interfering and the PV interacting RNAs. The psi is actually peculiar to plants, and the pi RNAs is like the homologue in animals. Okay, so the pi RNAs will perform the same function psi RNA does in animals, but psi does its work in plants. Okay, so what they do is they help protect the integrity of plant and animal genomes. Okay, so they actually prevent the spread of transposable elements to other chromosomal loci. Okay, so I'm sure you know what transposons are, or transposomes. Those are like nuclear material that can move from one place to the other. So especially when that particular DNA is toxic, okay, or let's say it can cause a mutation, or it can cause harm. So these these RNAs prevent the spread of these transposable elements, okay, from moving to other chromosomal loci. And we have this diagram showing like the effect of using um, the effect of repressing gene expression or degrading RNAs. Okay, so we do this using two proteins, the RISC and the RIT. I don't know if I still remember the full meaning of this, but the RISC is like RNA something something silencing complex. Okay, and this is RNA induced translation transcription something, but you can look that up. Okay, I'm sorry I've forgotten that. But what we do is we use the RISC complex, okay, with other proteins. I'll go through the steps to degrade RNA. Okay, so this is to prevent translation at all. So translation won't happen at all when we are using the RISC complex. Okay, and when we are using the RITs, RITs, we are repressing transcription. Okay, so transcription cannot move on. Okay, so let's look at each one of them individually. So first we have your long double strand RNA. The double strand RNA simply means like a folded RNA. Okay, so you use a dicer to cut them into different fragments. Then of course we can work with the double stranded RNA. So the small fragment are the siRNAs. Okay, so after dicing we get siRNA or small interfering RNAs. Then we make them, or we denature them, making them single strands. Okay, so Argonaut and other proteins, when we add our siRNA plus our RISC proteins, so first we add Argonaut and other proteins plus the RISC protein, the ones we are using to degrade your RNA. When you add the RISC proteins to the Argonaut, you and the siRNA comes in, it forms an activated RISC complex, right? So the Argonaut RISC proteins plus the siRNA forms an activated RISC complex. So the function of the siRNA is just to provide a complementary, like a sequence that will help it identify a particular place on the RNA you want to degrade. So let's say this is the RNA we want to degrade. The siRNA will be complementary to a particular sequence on the RNA. So it helps the activated RISC complex to bind to the RNA so that it can degrade it. Okay, so after degrading, the activated RISC complex will leave. Okay, and the initial steps is similar to the RISC. In the RIT is similar to the RISC. Same way, the RIT protein will bind to the will come together with the Argonaut and other proteins, and when the siRNA comes in, it becomes an activated RIT complex. Okay, so in this case, the what do we call it? This is like transcription occurring. Okay, so on your DNA, you realize it's like a polymerase, and your mRNA is being transcribed. So what happens is, with the activated RIT complex, it will bind to the mRNA being transcribed, okay, and it will recruit HMT and DNMT, okay, histone methyltransferase and DNA methyltransferases, and this methylate certain point of the mRNA, and this represses transcription. It prevents the mRNA being transcribed further. Okay, so this does not degrade the RNA, okay, but stops or represses transcription. Okay, so another form of these functional RNAs are the long non-coding RNAs. You can call them NC, that means like non-coding, or LNC, long non-coding. So this one, not much is said about it, just that what it does is not really documented, so its function is currently unknown. But there is a table here showing different types of long non-coding RNAs and what each one of them does. So what Doc actually mentioned in class was a cyst RNA, okay, that plays a role in regulating X chromosome inactivation. So you realize in females they have two X's, like two X chromosomes. One has to be inactivated and one has to be active. So it is the cyst long non-coding RNA that the cyst long non-coding RNA actually plays a role in inactivating one X chromosome, okay, and so the cyst, and it's like when you're reading from backwards like T6, okay, so the T6 will be like a regulator of the cyst, antisense regulator of the cyst. So what do we mean when we say an RNA is constitutive? Okay, RNA constitutive when it is continuously synthesized. Okay, so RNAs like mRNA, tRNAs and rRNAs and even snRNAs are always needed. Okay, so they are always being synthesized in the human body. But we have facultative. I don't want to use the faculty, it's not actually mentioned, but those are like induced RNAs, those that are synthesized upon requirement. So when they are needed, that's when they are synthesized. Okay, and those are like the miRNAs, silencing the small interfering RNAs, the pi and the long non-coding ones. Those are not constitutive. Okay, they are only synthesized when needed. And okay, let's talk about RNA processing. So RNA processing actually can occur post-transcriptional or co-transcriptional. Okay, so something like the capping, for example. So there are different, there are multiple modifications that we do to the RNA that you are transcribing, okay, or the RNA that you are synthesizing. So it is like we have splicing, capping, there are base modifications, but those are not post-transcriptional modifications. So the last one we do is actually the tailing, poly A tailing. I don't know if there are any others. Actually, they are, but those might be minor. So the first one that is done is the 5' capping. Okay, so we add a cap. Okay, it's actually a group, a chemical group that functions to protect the 5' terminal. Okay, and the cap consists of 7-methyl guanosine residue. So mostly you see it being written as MG7. So wherever you see MG7 on the DNA molecule, it's actually, or RNA molecule, it's actually the cap. Okay, and it is done at the 5' end. So when you are synthesizing or when you are transcribing an mRNA, the 5' end is synthesized first. Okay, so to prevent other the cell from degrading the mRNA that has been transcribed, we protect that end with a 5' cap. Okay, and that cap consists of the 7-methyl guanosine residue. So the cap is actually linked to the 5' end by 3 phosphate groups. Okay, so this diagram shows. So you realize this is your mRNA being synthesized. This is the 5' end, and this is the 7-methyl guanosine. It is linked to the 5' end by 3 P's. Okay, 3 phosphate groups, and 5' cap of most RNA is monomethylated. That's why you see only one, like 7-methyl, but in some RNAs, like the small non-coding ones, they are trimethylated. So example, it can be like 5, 6, 7 trimethyl. I'm not saying this, but that's an example. Okay, but in most mRNA, they are monomethylated. The cap has two functions. Okay, what it does is to protect RNA from degradation. okay, whilst the transcription is ongoing, and also it is required for translation. Okay, so when the mRNA moves outside the nucleus, it will be required for recognition and stuff before translation of that particular mRNA can occur. Another processing, RNA processing we do is polyadenylation. This is the addition of poly A tails. Okay, so adenylation is you are adding adenine groups. Okay, but poly, so plenty of them. So that will be at the end. So it is 3' polyadenylation. So RNA elongation continues until a particular signal or a particular sequence is reached. Okay, so when the particular sequence is reached, it sends a signal that this is the end. Okay, so when the end is sensed, then we add the poly A tail. Okay, so someone will ask, what is this sequence that will give a signal? The sequence is either an AAUAAA or AUUAAA, right? So when you are transcribing a particular mRNA and you come across, so when you are synthesizing, then you realize you are synthesizing AAUAA, then you realize, nah, I'm coming to an end, I should stop. Or you come across AUUAAA, okay, it will mark the end of the 3' end, and it will trigger the addition of a poly A tail. Okay, so the enzyme recognizes this sequence and cuts off the end of the RNA about 20 base pairs further down. So when this conserved sequence are reached, I say, oh, so I can't do, so as we just stop. So when they reach here, they just transcribe like about 20 bases further down, then they stop. Okay, then the enzyme that has the poly A tail, which is the polyadenylase, will bring in the poly A tail and add it to the tip of the RNA. Okay, so the AAU sequence of the mRNA is called the polyadenylation signal. So that's what triggers the polyadenylation or the addition of the poly A tail. And the adenylation requires a protein complex. Okay, so whatever enzyme, for us to add the poly A tail, we will need a protein complex, and that protein complex contains an endonuclease. Okay, and an enzyme that will add the polymerase. So the endonuclease will, of course, we know what an endonuclease does. It will cut. Okay, and the poly A polymerase is what will be adding the poly A tail. Okay, so the poly A polymerase or PAP, so the endonuclease will cut and the poly A polymerase will add the poly A tail. Okay, so this is how it works. This is a protein complex. Okay, so it has so many subunits. Okay, but now we are just focusing on the endonuclease and the PAP subunit. Okay, so the poly A polymerase adds nuclease residues. These are the AAAA, right? So it adds so after it adds, then the other subunit of the complex will dissociate. Okay, even including the poly A nuclease itself, it will leave. Okay, but there are poly A binding proteins as well, so those ones will remain intact on the poly A tail, but that's not what we are focusing on. What we have to focus on is what does the polyadenylation, and it is the protein complex that has both endonuclease activity and polymerase activity, which is the poly A polymerase. So another RNA processing that we do is intron removal or splicing. Okay, so now let's talk about the nitty-gritty. I'm sure we already know what splicing is because we've discussed it in the previous lecture. I'm not going to talk about this. So in splicing, we also have another type of splicing called alternative splicing. Okay, so I don't know if I should go through the introduction. You let me go through. So humans have about 21,000 protein coding genes. Okay, so take note of the number 21,000, but yet we have over 100,000 proteins in our proteome. But we say genes, specific genes code for specific proteins. So if so, why don't we have just 21,000 genes, but instead 100,000 proteins? Hey, why don't we have 21,000 proteins but 100,000 proteins? That means something is happening in the background, and a particular gene is coding for more than one protein. Okay, but the same gene without any modification cannot code for more than one protein because it's the same sequence, right? So what happens? And that's when alternative splicing comes in. So if you look at Sorry for that reminder. Okay, if you look at, I'm not showering though. I'm busy. I'll shower later. Um, so whatever we are going to talk about, the alternative splicing thing. Okay, it actually occurs in like exclusively in humans, but it is rare in plants. Okay, so let's look at alternative splicing. Let's say this is like a prime an mRNA transcript. Okay, so it has like numerous genes, and we have with introns and exons. Okay, so we have to splice it. Okay, so there are different ways you can splice this, like combining different genes. So we have 1A, 2A, 2B, 1B, 3, 4, 5, 6A, 6B. So let's say in let's think about 3. Let's just use 3 examples so that we can move faster. If we take this smooth muscle and the striated muscle DNA or mRNA, you realize even though they all stem from the same mRNA strand, they don't have the same genes. They might have they might have similar genes, but not all their genes are equal. So you realize they all have 1A, but the striated muscle does not have the 2A gene, but this one does. This one has this one does not have. So alternative splicing simply means different combinations of genes, okay, from the same mRNA transcript. Okay, so we are just combining different genes in order to produce different proteins. Okay, so that's why even though humans have 21,000 proteins, coding genes, we have over 100,000, like in excess of 100,000. That's like 121,000 proteins in our proteome. Okay, so one particular mRNA, if we were doing one gene, one mRNA to one protein, then it would have been this strand has to code for one particular protein. But no, you realize it's coding for 1, 2, 3, 4, 5, 6, 7, 8, almost 10, right? And this is as a result of alternative splicing. And I'm sure you know that snRNAs participate in exon splicing. As I said, they form a part of this spliceosome. Okay, so one thing we have to know, I'm sure I've discussed this, the intron exon junctions, but let's go through them again. So I, as already discussed, there are some specific sequences. When you realize this is where an intron starts and this is where an intron ends. Okay, so we might be given like numerous sequences, like letters, and just to fish out an intronic sequence or an intronic region. Okay, so what should we be looking out for? You should be looking out for a donor site, an acceptor site, and a branch site or a branch point. Okay, the donor site is the 5' end, acceptor site is the 3' end. Donor site is actually, in mRNAs, it starts with GNU, in DNA to be GT, and the acceptor site is strictly A gene. Okay, and this is called a GNU A gene rule. But minor introns do not follow this rule. So you realize there will be some introns that will not have that will not be following this rule, but major introns do follow this rule. Okay, so I think we've already mentioned the nucleotide sequence for the branch point. It is UAC, right? So that one is a bit conserved. So wherever you go, you are supposed to see that one to know that that's the branch point. Okay. And one thing about the splicing apparatus or the spliceosome is that it is not tissue specific. So the same spliceosome can splice different mRNAs in different genes or different cells. Okay. Okay, I think I've also mentioned this, that snRNAs are part of the spliceosome. Okay, and how they form part of the spliceosome is they are component of a particular component of the spliceosome is called the ribonucleoprotein. Okay, and the snRNAs are part or is a component of the ribonucleoprotein complex. Okay, so what the spliceosome does is it has protein component and RNA component, and it uses the RNA component is actually the snRNAs. Okay, it uses the snRNAs or the RNA component to fish out for specific part or the intronic regions so that it can bind to them and cut them. Okay, so ideally we can put it this way, that the snRNase component of the spliceosome are complementary to the consensus sequence at the splice junctions. Okay, and this makes it able to bind to the intronic regions. Okay, so when the spliceosome is coming in, it knows where to bind and where to cut because it has snRNAs, and these snRNAs are complementary to the target sites. Okay, so the SNRPs, there's small nuclear ribonucleoproteins. Okay, conserve blah blah blah, recognize blah blah blah. The SNRPs are complexes of proteins and about 1 to 5 snRNAs. Okay, so when about 1 to 5 snRNAs and proteins come together, they form small nuclear ribonucleoproteins. Okay, and examples are U1, U2, U4, U5, U6. There are 5 of them, but there is no U3. Okay, so the U1, U2, U4, U5, and U6 are what actually plays that's the splicing in mRNA. Okay, and we will look at how it is done. And the spliceosome is the large machinery, okay, of all the snRNPs, okay, doing the splicing. So how does the spliceosome interact with the mRNA? So I don't know, I'm not sure we've talked about the CT detail of the mRNA strand being transcribed, but those who did this particular course in third year, they should remember the CT detail. Okay, but I'm not going to go into the CT detail. Okay, but the spliceosome actually interacts with the CTD of the RNA polymerase 2. Okay, so that's the part of the mRNA it attaches to. Okay, then it can use its snRNA component to bind to the intergenic regions and do the splicing. So the U1 and U2 are the first proteins or the first snRNPs to be recruited. Okay, and what they do is they help the spliceosome to align to the splice site. Okay, so when they are recruited and they align the spliceosome, they will recruit the U4, U5, and U6. Okay, so this is the diagram. So you realize this will be your pre-mRNA with your exons and intron. So U1 and U2 comes in. The U1 and U2 come in. Each one of them can bind to any site, but in this case, let's say U1 binds to the donor site. So one will have to bind to the donor site and one will have to bind to the branch point. Okay, so any one of them can bind to the donor and anyone the other binding to the branch. But let's say U1 binds to the donor site and the U2 binds to the branch point. Okay, when it does so, we say it is helping the spliceosome align itself. Okay, or it is, let's see, it is showing it the way. Okay, so that's where you are supposed to come. So when they come in, so when they bind, they recruit the other snRPs U4, U5, and U6. Okay, and this will direct cutting. So when the U4, U5, and U6 come in, U1 and U4 will be released. You realize the U2 is still bound to the branch point. So the U1 and U4 will go away, leaving U6 and U5. So actually, what this does is it helps cut the first site. Okay, the removal of the U1 makes the other complexes cut the U point, the GU point, sorry, which is the donor site. Okay, and the donor site will be then form a lariat. What's it called? A cowboy's lariat. Okay, that's a loop. It forms a loop, and the shape is a cowboy's lariat. Okay, and this cutting is called a transesterification reaction. Okay, so splicing actually consists of two transesterification reaction. The first cutting is the first transesterification, and the second cutting that occur at the and the other exon intron region is the second transesterification. So when the first one is cut and the loop is formed, the intron or the exon that was cut first will go and cut the other end at the acceptor site. So let's look at the chemistry, and this will make it a bit easier. So what's happening here is the GU region, of course, nucleotides, the backbone of nucleotides are the phosphate groups. Okay, so the phosphate back groups are going to be cleaved. So the exon is bound to the GNU by a phosphate group. Okay, the exon is bound to the GNU by a phosphate group, and you have to cleave the phosphate group. Okay, so you cleave the phosphate group at this end, and the exon will have an O to its name, but this time around the phosphate, instead of having 4 O's, will have 3 O's bound to it. Okay, and the exon will be given an H because the O will make it reactive, so it will be exon 1 and OH. Okay, then the phosphate group, because it will be unstable, it actually needs 4 O's to be stable. So what it does is it will move here to the branch point. Okay, the branch point has an OH on it, and the H will leave. Okay, and the O, it will donate its O to bind to the this phosphate that's lacking an extra O. Okay, mind you, this phosphate, because it lost its O to the first exon, it has 3 O's now, so to go here and bind to the O at the branch points. Okay, so that's what happens here. Now the phosphate has 4 O's. So at this point we see it has looped over here in this diagram, forming the cowboy's lariat. Okay, so the exon over here with its O comes to the acceptor site. Okay, using its O, because it is reactive, it will cut or cleave the other phosphate group at the other exon end. Okay, and bind with the other exon. Okay, so the exon 1 and exon 2 will be joined together, and what happens is that now you realize you have only 1 O here. Okay, so that's why let's just see the H that left the branch point will be given to this one, the H inside, so that it won't be unstable. Okay, so realize the H inside will now be OH, and this is the lariat intron or the looped intron, and the exons will be linked together by a phosphate bond. Okay, so that's how splicing is done using the SNR RMPs proteins. Okay, so splicing can also be regulated by enhancers and silencers, and these will be called, because they are enhancing or silencing splicing, you can call them exonic and intronic splicing enhancers and silencers. Okay, so so in alternative splicing, right, you know that, so let me refer you back to this diagram. You realize in all this alternative spliced transcripts, okay, you realize each one of them have a particular sequence that remains constant in all the proteins transcribed. You realize all the strands have gen 3, 4, 5. All of them have 7 and 8 as well. They all have the 9. No, this one does not have 9. So let me guess. It is 3, 4, 5, 7 and 8. They all have these proteins. So we call these ones conserved or the constitutive. These ones are called conserved constitutive exons. Okay, these exons are constitutive exons, and the ones that alternate, that will be alternating between different proteins, are the alternative exons. Okay, so it is said that sequences surrounding the alternative exons are more conserved evolutionarily than sequences flanking constitutive exons. Okay, so the exons that are constant in all proteins emanating from a particular mRNA transcript are not evolutionarily conserved than sequences surrounding the alternative exons. Okay, so we can have splicing enhancers and splicing silencers, okay, regulating splicing. Okay, and actually there are proteins that bind to specific enhancer regions and silencing regions that will do this regulation. Okay, so let's look at this diagram over here. You realize there are different protein binding sites, okay, and different proteins are going to bind to it to either enhance or silence. So in this case we are looking at splicing regulation, so either enhance or silence splicing. Okay, so so realize in general SR proteins bind to exonic splicing enhancers. SR proteins are, so SR is actually 2 amino acids. S is serine, R is arginine. So serine arginine rich proteins. Okay, so serine arginine rich proteins. What am I saying, bro? Charlie. So in general, SR proteins will bind to enhancers. Okay, and this is going to enhance splicing. But there are silencers as well. The HNRP. These are heterogeneous nucleotide ribonucleoproteins. Okay, and in this case, the two families that are going to talk about, or you are going to see in the diagram, are the A and B families. Okay, they bind to silencer regions to silence splicing. Okay, and that's all there is about in this slide. That's all there is about splicing, regulation of splicing. So the splicing we talked about is actually in mRNA. So now let's look at splicing in tRNA and splicing in rRNA as well. Okay, and if you remember when we discussed the 3 or 5 types of prokaryotic RNA polymerases, okay, you realize we said RNA. Hey Charlie, am I hungry or what? No, I'm not from sound. So RNA polymerase 1 actually transcribes rRNA. Okay, RNA polymerase 2 transcribes mRNA, and RNA polymerase 3 transcribes tRNA. Okay, I'm sure you remember that. So in the transcription of tRNA, of course, the polymerase 3 will terminate transcription in a poly-U sequence. Okay, this is not like the termination of transcription of tRNAs is not like termination of transcription of mRNAs. Okay, in termination of mRNAs transcription, we add poly-A tails, but in tRNA transcription, transcription is terminated by in a poly-U sequence. Okay, and when this is done, the tRNA that's produced is called a premature or a pre-tRNA. Okay, and tRNA splicing occurs by successive cleavage and ligation reactions. So you have to, just like in mRNAs, you cleave the introns, then you ligate the resulting fragment, the exons. Okay, so what does the splicing? I'm sure you know what does the particular RNA component of the splicing machinery that does the splicing. Of course, it is the snRNA. But now we are not really mentioning it directly. Okay, so the protein that does the splicing is an endonuclease, and the name is an RNase P, ribonuclease P. Okay, so you should know that the snRNA, I don't know, some way somehow should be part of this, because we've already established that the snRNA is involved in the processing of rRNAs and tRNAs. Okay, so the RNase P, which is an endonuclease, is what does the splicing. Okay, so this is how it actually looks like. So this is the premature tRNA, and the red portion is the intron. Okay, and the violet or the purple portion is the exons. Okay, so you cleave the exon away. Okay, then you create some unusual ends, and these unusual ends will have to be modified. Then we can join the two portions to form the clover leaf tRNA or the fully matured tRNA. So let's look at what happens after the RNase P cleaves or splices the tRNA. We create two half tRNAs with unusual ends. The reason why we call these ends as unusual is because normally should I call it a nucleotide or any nuclear material, right, a nuclear strand, should have two ends, and those two ends should be 5' or 5' phosphate, like a 5p. So the 5' end should end in a phosphate, and the 3' end should end in an OH. Okay, but these two tRNAs that we've gotten ends in 5' OH. Instead of 5' phosphate, it is now 5' OH, and the other end is 2', 3' cyclic phosphate. This is very unusual, so it prevents further reactions. So what do we have to do? We have to modify these ends so that we can get the 5' P or the 5' phosphate and a 3' OH, so that we can join them together. Okay, so how do we convert OH to phosphate? We just phosphorylate it using a kinase. Okay, the reason why we call it polynucleotide kinase is because we are working with a nucleotide, and it is numerous nucleotides. That's why polynucleotide kinase, and we know kinase is an enzyme that has phosphate groups. Okay, so we just phosphorylate the 5' OH for it to become 5' phosphate. Okay, and with the cyclic phosphate, what we do is we have to open it because it's cyclic. You have to open it first before we do any other treatment. So the opening is done by phosphodiesterase. Okay, and when we open, we get 2p and 3OH. This is very ideal because this is what we wanted. So we will just get rid of the 2p end. Okay, so we get rid of the 2p end using a phosphatase. Okay, then we join the 3' OH and the 5' P, okay, together to form our mature tRNA. Okay, and in rRNA as well, splicing in rRNA also occurs a bit different from the previous two we've talked about. So this one first makes use of endonucleases. Okay, so rRNA in these cases are like linear strands, and we all know they have like different subunits. 18S, the small subunit. In oh, sorry, I'm coming. In in eukaryotic RNAs, okay, we have the 18S, 5.8S, 5S, and the 28S. Okay, in prokaryotes they have the 5S, 23, and the 16. So now let's assume, it's not assuming, the topic is actually about eukaryotic RNAs. Okay, so obviously we're dealing with the eukaryotic RNAs. And if you remember, when we're doing the transcription, the polymerase that took charge of the transcription, you realize the smaller subunit of the rRNA wasn't transcribed by RNA polymerase 1. Okay, so RNA polymerase 1 will transcribe all the subunits of rRNA except the small one, the 5S. Okay, so that one was actually taken charge by RNA polymerase 3. Okay, so we will let's come back to the splicing. We will need endonuclease. Endonuclease cleaves inside a strand. Okay, so endonucleases will cleave within. So endonucleases that are specific to the intronic regions. Okay, so they are going to cleave the intronic sequences, the ITS, intronic sequences. So when they cleave, and the fragments that we get, we use exonucleases to cleave them. Okay, and this will result in the individual rRNA subunits that we need. Okay, that's a bit straightforward as well, but there are some other component of the SNORNAs that, and this actually let's look at different parts of the SNRNAs and what they do in processing of these rRNAs. Okay, so the SNRNAs have a particular group called a CD group. Okay, and this CD group are used to add methyl groups, okay, to the 2' position of the ribose on rRNA. Okay, so this would be like the CD group. Okay, so it uses the SNORNA is actually complementary to the rRNA that's to be methylated, okay, or the region to be methylated. So it binds to the RNA that's to be methylated and use its CD group to methylate it. There are other components of the SNRNAs like the H-ACA, or I call it HACA. Okay, the H-ACA group is actually made of two hairpin loops, okay, with two consensus sequences, or how do we call it, conserved sequences. Okay, the ANANA and ACA way. But what it actually does is it just converts non-bound uridine. So when what it does is it actually binds to the rRNA, and any U that's not bind to anything, that's not bound to anything, will be converted to pseudouridine. Okay, so these are just portions of SNRNAs that does modification on rRNAs. Okay, so the CD group of SNRNAs that's methylation, and the H-ACA group that's conversion of uridine to pseudouridine. Now let's talk about self-splicing introns. Okay, so before 1981, probably they saw RNAs as just nuclear materials or nuclear biomolecules and not as enzymes. Okay, but someone realized that there was a particular organism, tetrahymena, that could use that a particular nucleotide in tetrahymena could cut itself. Okay, the intronic sequence in that nucleotide could cut itself without any protein. But we know proteins by then, they knew proteins were like all enzymes were proteins, right? So any biochemical reaction is actually catalyzed by an enzyme. So without the addition of a protein, they weren't expecting the nucleotide to the intronic sequence to be able to splice. Okay, but they realized it could splice on itself, and they termed these introns as self-splicing or auto-splicing. Okay, so this led to different propositions. It's not really okay. Initially they were propositions, but now they are actually certified theories. Okay, that there are some RNAs that have enzymatic activity as well. Hence, some enzymes are RNAs. So, another proposition was this man Sidney Altman discovered that RNase P, okay, which is involved in cutting the pre-tRNA, okay, is actually the catalytic portion of the catalytic portion of the RNase P is actually its RNA component and not the protein component. Okay, so this is just reaffirming the fact that some RNAs can serve as both nuclear materials and enzymes. Okay, and they also found, another person also found that in the spliceosome, in the spliceosome, the catalytic activity is actually done by snRNAs and not the protein components. Okay, and this led to different propositions like RNAs must have been the genetic material in the first cells, but I don't believe in this evolution bullshit. I'm just learning for exams. So this led to the word ribozymes. Okay, that there are RNAs that can act as enzymes and nuclear materials at the same time. Okay, so what our focus is actually on self-splicing introns or auto splicing introns. Okay, so within this we have two types of the self-splicing introns: the group 1 introns and the group 2 introns. So let's talk about group 1 introns. Please, if you are sleeping, wake up. If you are dozing off, open your eyes. So group 1 introns are the splicing actually in a diagram. It actually looks similar to the splicing in mRNAs. Okay, but the mechanism is actually not the same. So what it does is it also requires or is made of two esterification reactions, just like in the splicing of mRNAs. Okay, but here you won't need any proteins, the U1, U2, U4, blah blah blah, right? So the required factors are just two metal ions and a guanosine nucleotide. Okay, this guanosine nucleotide is actually it has a OH group. Okay, so the OH group of the guanosine nucleotide will first the OH group of the guanosine cofactor will actually cut the first exon intronic junction, okay, at the 5' end. Okay, so when it cuts in the you can just try remembering the diagram that was in the mRNA. You realize the exon over here will have an OH. Okay, and to use that exon, that's the first exon that's cut, which is exon A, you will use its acquired OH to cut the second exon intronic junction, okay, which is the acceptor site. Okay, to cut that one, and the two exons can be joined together, and the resulting intron can circularize. Whatever happens to the intron next, I'm not interested, right? But the group 1 introns can do other things like moving from one target to another, and we will come to that. So the group 1 introns form characteristic secondary structure. So when they are cut, it's not even only when they are cut, but they can be linear and they can be folded. Okay, so and this is actually regulated by things we call the ribose switches. Okay, so whether they are forming a secondary structure or they are linear, it's actually controlled by things we call ribose switches. So what they do is they can form a secondary structure, and this secondary structure contains 9 duplex regions. So you can see it in this diagram. You have like 1 2 3 4 5 6 7 8 9, like 1 2 3 4 5 6 7 8 9. This is supposed to be another one, but it's not showing, so probably. So, and these duplex regions are named PPPP. Okay, and in these duplex regions, P3, P4, P6, and P7 have the catalytic activity. Okay, so it is only when, in this case, in the group for group 1 introns, only when the intron folds this way, that's the self splicing can occur, or the automatic splicing can occur. Okay, because when they do not fold, the catalytic component of the intron won't be formed. But when they fold, the P3, P4, P6, and P7 will come together to form the catalytic center. Okay, and that can induce the cutting of the introns and stuff. So you realize when the splicing started, the intron had folded already. Okay, but in the normal splicing, the introns were straight. Okay. So some group 1 introns, as I said, can move from one DNA molecule to another. Okay, and what they do is they encode proteins, and these proteins are endonucleases. Okay, so they can translate an endonuclease, and the endonuclease will move to wherever they want to go. That will be like their target site, and the endonuclease will go and cut there, and then the intron will replicate itself, or a copy of the intron will be made. Then it will just insert itself into the new site that has been cut already by the endonuclease. So this is one of the reasons why we were actually asked to redefine our definition for introns, because introns are said to be non-coding regions. And but one thing about this is that this does not apply to all introns. Okay, these are just some selected few introns that does this, or that bro, for that do this. Another term called intron homing. Okay, intron homing is actually about, so there's this movement is called transposing. So transposing element actually uses this mechanism as well. So the intron moving from one place to another is called like it being transposed from one DNA molecule to another. Okay, so there's something we call intron homing, and this refers to the specificity that ensures the intron will perpetuate itself only by inserting to a single target and not any elsewhere in the genome. Okay, that's what we call intron homing. And the second type of self-splicing introns is the group 2. Okay, this is a bit different from group 1s. So what they do is they code for a protein, and that protein is multifunctional. It has different domains, or yes, different domains with different functions. Okay, so when they code for this protein, the protein should have a reverse transcriptase activity, matrix activity, RNA, an endonuclease activity, and it also should have a DNA binding motif where DNA can bind to, or where it can use to bind to DNA. Okay, but this is not really a function. Okay, the main functions are the reverse transcriptase, matrix, and the endonuclease. So endonuclease is when the part of the protein that will use to cut the target site. Okay, and the reverse transcriptase is used to generate a DNA copy of the RNA. So I'll actually use the diagram to explain all of this. And the matrix is to assist the RNA to fold into a matured structure. Okay, so first, this would be your premature DNA strand, like your DNA strand with exons and introns and everything, but it will be translated, okay, into your pre-mRNA. Okay, so what this does is it will code the only the intronic sequence part, the intronic region will code for a protein. Okay, and we know the protein has the endonuclease. Okay, before we even go there, after coding for the protein, you cut the intron. Okay, then you join the exons. The exons are the purple parts. So that's why we've joined to get our matured mRNA. And this is the intron, the red one. So the intron will coil up and somehow attach itself to the encoded protein. Okay, so just like the group 1 introns, they can also move from one DNA to another target site. So the protein, okay, so it's like IEP is called the intron encoded protein or something. Okay, so the IEP protein will move to the target site wherever it's supposed to go and use the endonuclease activity part of it to cut the target site. Okay, then it will insert the mRNA. Okay, and it will use its reverse transcriptase to generate, it will insert its mRNA, sorry, so it will insert the intron. We know the intron is from an RNA, so it is an RNA. It will insert the RNA and use the reverse transcriptase to generate a DNA copy of the RNA. Okay, because it is inserting itself into a DNA, but it itself is actually an RNA. So it has to convert the RNA to a DNA sequence, and converting RNA to DNA is like backwards something, because normally we move from DNA to RNA. So when you're moving from RNA to DNA, that's why we call it reverse transcriptase, because transcription actually moves from DNA to RNA. So moving from RNA to DNA is reverse transcription. Okay, and the enzyme is a reverse transcriptase. So after this, after doing everything, the matrix part will assist folding of the intron. Okay, so in part 2 we will discuss mRNA decay and degradation. I'll see you in the next one.